Hi, this is Diana. Today we're going to talk about the blood agar. The blood agar contains general nutrients and 5% sheep blood. Some organisms produce the exozyme called hemolysis, which are able to destroy the red blood cells. Hemolysis is the destruction of the red blood cells. And there are 3 different types of hemolysis, which are alpha-hemolysis, beta-hemolysis, and the gamma-hemolysis. Blood agar is a differential agar. To determine the hemolytic capability of an organism, which means to test the ability of an organism to break down the red blood cells. And for the first plate here, we were using Enterococcus faecalis. We can see the hemolysis which were produced by Enterococcus faecalis partially destroy the red blood cells and with a greenish zone. And this is alpha-hemolysis. Here I'm going to explain why there is a greenish zone around the colonies. And blood cells contain the hemoglobin. When the blood cells are destroyed, the hemoglobin gets broken down to the heme molecules and the globin polypeptide chains. The polypeptide chains have been broken down to the amino acid, which can be used again to make new proteins. And the leftover heme molecules are toxic to the body, so must be broken down. And there is an enzyme called the heme oxygenase, which can convert the heme molecule to biliverdin, which is a green pigment molecule. So this is the reason why alpha-hemolysis presents a greenish zone or green halo on the blood agar. For the next plate, we were using Streptococcus pyogenes. So we can see the hemolysin which was produced by Streptococcus pyogenes completely destroyed the red blood cells. And because the colonies completely lost the color and becomes transparent. You will be able to see this transparency when you're holding up to a light in the lab. And for the next, we are using E. coli. We can see it appears as simple growth with no change to the medium. If the organism does not produce hemolysis, the red blood cells will not be broken down, so this is called gamma-hemolysis. Here is a good picture to help you distinguish alpha-hemolysis and beta-hemolysis. And we can see some colonies completely lost their colors and become transparent zone, which are the beta-hemolysis. However, some others are partially transparent here, which are the alpha-hemolysis. So this is such a good picture to show you how different between alpha and beta hemolysis. Thank you for watching.